Hello, everyone. Welcome to today's talk on ALS clinical trials and the NEALS Consortium. I'm Mary Ann Mertz. I'm your moderator for today's talk. I'm one of the nurses from ALS United, Greater New York, and I work out of the clinic at Rutgers Robert Wood Johnson in New Jersey. After today's talk, Dr. Andrews will entertain questions. So if you have any questions, please place them into the Q&A box. And I'm really honored to be able to introduce to you Dr. Jinsy Andrews. Dr. Andrews is currently the Director of Neuromuscular Clinical Trials at Columbia University. She has been actively involved with the NEALS Consortium for the past 10 years and has participated in many NEALS clinical trials. She also serves as co-chair of the Upper Motor Neuron Disease Committee of NEALS. And prior to her current position at Columbia University, Dr. Andrews was the head of Neuromuscular Therapeutics and Senior Director of Clinical Research and Development at Cytokinetics Incorporated, overseeing international clinical research development on ALS and North American clinical research development in spinal muscular atrophy. Prior to Cytokinetics, she was the director of the Clinical Trials Unit and co-director of the MDA ALS Clinic at the Hospital for Special Care in Connecticut and assistant professor of neurology at the University of Connecticut. She completed her fellowship training in neuromuscular disease and clinical neurophysiology at Columbia University and subsequently held a faculty position as assistant professor of neurology while actively participating in clinical trials and caring for patients with ALS and other neuromuscular diseases at the Eleanor and Lou Gehrig MDA ALS Center at Columbia University. She is board certified in neurology, neuromuscular disease, and electrodiagnostic medicine. So, Dr. Andrews, I turn it over to you.
Thank you. That was super generous introduction. And I see why I'm tired, but I am really excited, and I really want to thank ALS United for inviting me today to give this talk. And I received a lot of feedback from people. So although the title says what it is, I'm going to try to address people's questions that I get in the clinic about current treatments, research, clinical trials, what can they do now, what can we look forward to in the future. So I'll try to kind of cover that. And hopefully, if I don't get to something that you were interested in, we can kind of discuss it in the Q&A session. So what I'll do is I'll just get started and just start from here. So hopefully you can see my screen at this point. And then hopefully it's in presentation mode for you, for all of you. If it's not, just pipe in and let me know. So today I'm going to talk to you first. I know there's some of you that are really familiar with the current treatment options, some of you that are new. And I just wanted to go over it because last year was very confusing and I still get a lot of questions. So the mainstay for treatment in ALS, and I'm just going to jump right in because we got a lot of stuff to cover because it's been an exciting year. So riluzole still remains the mainstay. This is the medication that has been here since the 90s. It was based on 2 clinical trials that were done, and it was on a dose of 50 milligrams, 1 tablet twice a day. And more recently, different types of formulations were developed to help alleviate some of the difficulties with swallowing that occurs in ALS. And so there was a liquid formulation, Teglutic, that was approved more recently, and also the oral film. And, you know, our community has asked the question, why even go back and reformulate riluzole? Shouldn't we just move on and get new drugs? And so many of us had gone back to look at the old studies and see whether there is a value here beyond what we hear. Most physicians cite a quote unquote 2 to 3 month improvement in survival compared to placebo, but there might be more to the story here about riluzole. So initially in 2018, the group out of the UK led by Amar Al-Chalabi had looked at the old clinical trials, and they developed a newer staging system. It's based on anatomy on where you're involved. So stage 1 is 1 region involved. Stage 2 is 2 regions involved. Stage 3 is 3rd region. Stage 4 is when you need a ventilator to help with breathing, or you need a feeding tube to help you supplement your nutrition. And stage 5 was death. So they went back and said, well, let's look at the old studies and see, instead of looking at survival, are we delaying transition to stages? And what they found is that they were able to delay, they were able to prolong people in stage 4. And there was some minor delays in stage 3 to 4, but really we couldn't assess because most people come to the clinic and say, hey, doc, if I'm early in my disease, I want to stay as early as possible. Stage 1, can you make sure I stay there? And it was hard to assess based on the studies that were done because people were allowed to have disease for 5 years or less. And so most of the people enrolled into those early riluzole studies after stage 1. Another study looked at existing clinical trial, and so there's clinical trial data that's in a database, and then there's real world data. So myself and a couple of the colleagues in ALS looked at the real world data because it's been around for 20 years to see what the survival benefit is in real world practice. And there were significant differences in people who took riluzole versus who never took riluzole. And the median survival was anywhere from 6 to 19 months, which was way more than what we saw in the clinical trials. But given that we saw that, Dr. Mitsumoto and his colleagues actually went to the clinical trial database saying, well, maybe it's because riluzole got approved, it's been available, and we're starting it earlier than when we did in the clinical trials. And sure enough, when he looked in the clinical trial database, the earlier you started riluzole, the more the benefit. The later you started riluzole, the less of a benefit. And this kind of is a running theme for realizing that we need to start, if we have any disease modifying therapy, we need to start as early as possible. If we want to start as early as possible, we got to get the diagnosis earlier. We got to get more access to clinics. And so I think that's the initiative in our community. And we're distributing a tool called Think ALS to general neurologists, general neuromuscular clinicians to identify symptoms, and hopefully that can speed up diagnosis so that we can implement these therapies earlier. Okay, so we have riluzole. The next story comes in at 2017 with edaravone, or what we call in the United States, Radicava. This was a 6-month study looking at the overall function over 6 months, and there was a 33% slowing of decline in this very restricted cohort of people who had disease less than 18 months and were rapidly declining. And so when they looked after the 6 month, this study gave everybody, so it was placebo controlled for the first 6 months, and then they transitioned everyone to treatment. And that was the open label extension. It's almost like what you might call a delayed start study. A lot of that has been done in Parkinson's disease. And what they found is that there was a delay to significant events, milestones of loss of function. And so based on that primary controlled study and the open label extension, it received approval in the United States, initially as an intravenous medication, but because of the challenges of giving intravenous medication to people living with ALS, we were fortunate for a bioequivalent study to be completed, and an oral form came in 2022. And that's an oral dose that you take 14 days for the first cycle and then 14 days off. And then the maintenance dose is 10 days out of 14 days and then 14 days off. The controversy comes last year. So another company, different from MT Pharma, so this is Ferrer in Europe, had a different formulation of edaravone. And this study was a large phase 3 conducted mainly in Europe. And they did not see the same benefits on overall function and survival. And so there was data that was presented in the European ALS meeting last June. And there were some key differences between the studies that were completed. The formulation in the US is something that is given orally in an oral suspension, and it is cycle dosing. The formulation studied in Europe are granules that are dissolved in water, and it's 100 milligrams, and you get a dose every day. It's manufactured differently, and more importantly, the clinical trial population was different. Does it raise questions about the efficacy of edaravone? I think there's still a conversation to be had, but the study did not impair the existing approval of Radicava or edaravone in the United States. And you might hear mixed feelings from physicians and researchers about edaravone and whether it is effective or not. There are still a lot of unanswered questions, and MT Pharma has presented some preliminary work in the preclinical space for effects on oxidative stress, and they are doing a study to look at biomarkers, and we need that information to kind of give you more confidence about what we expect when we use edaravone. The third medication that created a little bit of ruffle in our community last year was Relyvrio. As everyone knows, it completed a phase 2, and looking at overall function similar to the edaravone study, it showed about a 25% difference between treated and placebo at the 6-month mark. And there was a full approval based on the effect on ALS-FRS on pre-specified analyses and confirmatory evidence that was obtained through open label extension. So that's a sign of FDA flexibility. That is not the usual or typical way FDA gives full approval for a drug. Usually you need 2 phase 3 clinical trials that are completed, that are placebo controlled. But they allowed full approval contingent on a phase 3 being completed, and that was the Phoenix study. The Phoenix study was completed, and it did not show the same effect as the phase 2 study. So this is the graph from the phase 2, this is the graph from the phase 3. So the company made an announcement last year that they were going to pull it off the market, but there are still data that needed to be collected and still reported out. Biomarker analyses were not completed at that time of the decision. Subgroup analyses were not completed. Survival analysis weren't completed at the time of deciding to pull it off the market, but it did come off the market and became completely off by October 2024. So we no longer have that in our tool set in the United States. And so we move on, and the most recent news came in the international meeting at Montreal for the International ALS MND Symposium, where the Japanese group did announce that they did a study on ultra-high dose methylcobalamin, which is a different formulation or unique formulation of B12 that's injected. And they looked at early stage ALS patients, and they did a placebo-controlled study and saw a significant difference. There was very little dropouts in this study, and they presented additional survival data this past December, which helped support this preliminary data that was called, the clinical trial was called JET-ALS. Based on this information, Eisai, I hope that's the way you say it, filed for an approval in Japan. So it received approval at the Japanese regulatory authority as another treatment for ALS. Although we can compound it here and B12 is available in different formulations here in the United States, it is not an FDA-approved medication yet. And I don't know, or I'm not aware personally of any plans for filing in the United States. So if people pursue this injected formulation, so typically it's 25 milligrams and sometimes 50 milligrams injected 2 times per week intramuscularly. Usually it's an out-of-pocket expense and you have to get it through a compound pharmacy. All right. If that wasn't confusing enough, let's go to more simpler topics like genetic forms of ALS. When someone has a gene mutation and it is something that is known to be pathogenic, it makes it simpler to understand what you need to target for treatment. But the complicated thing about ALS, and this slide, actually there could be a whole talk for 1 hour just on genetics and ALS, because it's more complicated than even this slide shows, because now we understand that there are genes that cause ALS, and we understand that there are genes that can put you at risk or modify disease progression in sporadic ALS. So there's different genes that are pathogenic that can be mutated and cause ALS. And I think that's what causes a lot of difficulty in kind of untangling even the genetic forms of ALS. But because we found different gene mutations that cause ALS, we understand some of the mechanisms that are not functioning properly in your motor neurons, right? So neuroinflammation, mitochondrial dysfunction, oxidative stress, excitotoxicity. So all of these gene mutations have provided clues as to what might be going on in the motor neuron and what we can target as mechanisms to help prevent the motor neuron from dying. And so our greatest success story is really tofersen and SOD1. So this is an intrathecal medication, gene therapy that we deliver. So you person needs a spinal tap. We give it in a bolus injection, and people have to get it in a monthly dose. But the clinical trial essentially showed that when SOD is mutated, it produces a protein, and we can measure that protein in the CSF or the spinal fluid, and we want to lower it. And so it was successful in lowering the protein, SOD protein, and also lowering what we call neurofilament, which is a protein we want to lower, because if it's elevated in your blood, it's a sign that there is damage to the structure of the motor neurons, if simply put. What we did see is that although we moved these markers, these biomarkers, it took a while for the clinical signs to show. So after the 6-month mark, which on this graph is the dotted line, afterwards we see the line separating for the treated, which is the red, and the placebo arm, for clinical measures in ALS. And there was a really great paper that was published in early this year in January from the group, Dr. Sean Smith and the WashU group, where they looked at real world evidence, and they treated people and measured quantitative muscle measurement, and they noted stabilization of disease in the real world setting. So tofersen can make a difference. It's hard to receive the treatment because people have to go into the centers and they have to get spinal taps and have the medication put through. But 2023, we received accelerated approval in the United States. 2024, Europe gave a conditional approval under exceptional circumstances. It's our first accelerated approval in ALS. And we actually have pushed the bar a little bit further, where we say, okay, well, we know SOD1, we know which mutations can cause ALS. If there are volunteers that don't have ALS and that we check for the mutation and we follow their neurofilament, could we potentially prevent ALS from happening if we have tofersen? So the study is called ATLAS. It's currently fully enrolled, and we're really thankful to all the participants and their families who have participated. I say hi to some of them because I know them personally, but they've been followed on a monthly basis. We're following neurofilament levels, and then if they hit a threshold, we start treatment with tofersen. If they develop symptoms and they don't hit the threshold, then they just get treated with tofersen in the Part D section. So we're anxiously awaiting the data from that study. With all that said, we still need better treatments for all ALS. And so the clinical trial landscape is growing. And there are so many different targets, as I mentioned before, and different ways we can approach ALS. Most of the treatments that we're trying to develop under clinical trials are things that are targeting commonly oxidative stress. Actually, the most common target is neuroinflammation right now. Autophagy, endoplasmic reticular stress, a variety of ways of targeting the actual motor neuron. But there's a couple of products that are in the pipeline that are targeting the nerve and muscle communication and improving the neuromuscular junction. That's newer therapies and newer targets that have just launched. And then, of course, we continue to develop cell therapies like T-cell therapies and mesenchymal stem cell therapies to modify neuroprotection. And so this is just a snapshot. It might be a little outdated. It's March. Every month I have to update this slide. The most notable difference, I've given talks here at this education summit, and actually last year to this year, the biggest difference on this slide is if you look at the pipeline, there's been a huge growth in phase 1, phase 2. One of the things that I did notice personally is that a lot of those compounds are being developed outside the United States, which may suggest some challenges in kind of launching those phase 1 trials efficiently here in the United States. One of the compounds there for HERV-K suppression, it was like so recent, this publication, the Lighthouse study in Europe did not show an effect with a treatment that was targeting HERV-K, so that it has been terminated. But the highlighted with a star means that it's actively recruiting. If it's highlighted in red, that means it's in the United States. And if it's got 2+ signs, it means that it's going to start here in the United States. So as you can see, we don't have a lot of drugs at the moment that are actively recruiting, but phase 1, phase 2 pipeline is still bigger than our phase 3 pipeline. I will note that there's a lot of compounds right now that are actually going to come into phase 1. We talk about gene therapy and we talk about familial or genetic ALS, but there's a number of compounds like XT150 in the phase 1 column. There's Leal Therapeutics that's developing a therapy, Trace Therapeutics that's developing a therapy to modify particular genes for sporadic ALS so that we can hope to modify disease progression. So gene therapy may not just be for familial or genetic forms of ALS. And if you move to the phase 3 side of things, ibutylast is a drug that is a fully enrolled phase 3 that's ongoing called Combat ALS. Masitinib, I haven't heard any updates. It was enrolling in the United States a couple of months ago, and we're waiting for some feedback from the sponsor. And then upcoming phase 3 in the US, there's Clean Nanoscience is developing gold nanoparticles called CNM-AU8. Pridopidine is being developed by Polenia, and Prime C, which is a combination of Cipro and Celebrex, and probably the most discussed investigational product in my clinic. These are actually conducting feasibility surveys and trying to launch in the United States late this year and early next year. So that's kind of where we're at in terms of small molecules. From there, we'll talk about cell therapies. And this slide used to be a tiny little slide I could actually put in with the other slide, but there's more cell therapies developing and a real big interest in T cells. And so autologous means it's cells derived from your own body. Allo or allogenic is cells that are derived from someone else. So there's several companies working on T cell therapy. So there's the allogenic T cells derived from umbilical cord blood that's banked at a bank. And Selenco's is trying to plan a phase 2 study. Hopefully Columbia will be a participating site in the future. Rappa Therapeutics has a phase 2 clinical trial that's actively enrolling. So the clinical trial is in Boston, but the EAP, if people are not clinical trial eligible, Boston and Hackensack both have teams that are enrolling. Koya Therapeutics is taking a different approach by giving you abatacept and IL-2 to stimulate T regulatory cell production. And then there's several stem cell projects that are still ongoing. So Cedars-Sinai Group in Los Angeles are still injecting stem cells, human neural progenitor cells, into the motor cortex. This is a phase 1 study, so we're really learning about safety and tolerability. And there's been several subjects that have gone through and have done okay, but they're still recruiting individuals in Los Angeles. And Brainstorm is still developing. They've gotten feedback from the FDA and a vetted protocol, and they're trying to launch their phase 3 in the United States. It's a second study after doing their initial phase 3 study, and that's Neuron and bone marrow-derived mesenchymal stem cells. Now, gene therapies also have grown in the pipeline. So there are, because antisense oligonucleotide has reached approval in other disease areas like spinal muscular atrophy, it is the most mature technology that we can apply to genetic mutations. So the FUS gene has an antisense oligonucleotide. It's currently on hold, but we anticipate it'll be open for enrollment. Tofersen, like I said, is being used in this pre-symptomatic individual study called ATLAS. AP101 is a humanized monoclonal antibody targeting these mutated SOD1 proteins, and it goes in there and tries to take it out of the nerves. And that's ongoing as well. ALN-SOD is RNA interference. It's a therapy that's being developed by Regeneron, and that's actively enrolling currently outside of the United States. And AMT-162 is being developed by Unicure. It is working on encoding microRNA, and it is actively recruiting. Columbia University is an active site. And so this AMT, I'll just highlight a few studies that are really in our greater New York area. So as I mentioned, AMT-162 is a gene therapy for SOD1, for people who carry the SOD1 gene that is causing their ALS. It is a viral vector-based gene therapy, and it expresses microRNA designed to knock down those mutated SOD protein. So Dr. Harms at our site is one of the investigators, the principal investigator, recruiting individuals for this study, and it's a multi-center study. Gene therapy is hot because we've got a lot of technology, antisense oligonucleotides, viral vector technology. The ASO therapy is really ripe for applying for different types of therapies. And so Dr. Schneider, our director at the Eleanor and Lou Gehrig ALS Center, has been federally funded for this project called Silence ALS. They've been working very hard on TAR-DBP and CHCHD10 mutations. There is ongoing efforts to kind of launch into the other mutations. And so if those mutations are things that are affecting you or your family members, Silence ALS may be an avenue. That's a collaborative project between Columbia and Lorem Foundation, the NIH, to kind of look at these ultra rare mutations and see if we can treat people and help their disease. A different approach in New York is the spinal cord associative plasticity for ALS. So this is a government-funded VA, partially VA-funded study through the Bronx Veterans Hospital, Bronx VA, James J. Peters. And Dr. Harrell is the principal investigator. And what they're doing is using brain and cervical spinal cord stimulation to see if they can they can encourage kind of change in the spinal cord and achieve better function. And so the current proposal is to improve function with this is electrical stimulation in the hand function by stimulating the nerves, the origin of the nerves, which is the cervical spine. So the eligibility factors, you have to be diagnosed with ALS, have some weakness of the hands, but not total weakness of the hands, because you have to do tasks after you have stimulation. And then, of course, no history of seizures or implanted devices, which can interrupt the electrical stimulation. So they are actively recruiting individuals, veterans and non-veterans, at the Bronx VA. Other studies that people ask about enrolling in our area, I mentioned the RAPA Expanded Access Program. So if you're not eligible for clinical trials and diagnosed with ALS and have a vital capacity less than 50%, you could be eligible, and Hackensack Hospital is the contact for that expanded access protocol. It takes time, and there is a significant number of people interested, but there's 10 centers across the country. And the newest expanded access program, RAPA and Ibutolast, are both federally funded through the NIH. This is led by Dr. Bjorn Oscarson at Mayo Clinic, Jacksonville. This also is an expanded access looking at using Ibutolast. So when I say expanded access, there's usually no placebo, no control arms. And it is to be conducted in parallel to the ongoing phase 3 study. And it'll be in 15 centers, and they target 200 people living with ALS that are not clinical trial eligible to enroll. In order to register, you have to go, it's a little bit different. Instead of clinicaltrials.gov, you have to actually go to Wide Trial, which is a CRO specialized in expanded access. And I put the link there, https widetrial.com. And when you go to the website, it looks like this. And if you're a physician, you can click for physicians, but if you're a person living with ALS or a family member doing it for someone, you click for patients and an interest form comes up and you put your data and they'll contact you with information on the study and where to go or who to contact. If you're someone living with ALS and also interested in participating in natural history studies, I think it's super important. And I'm making a pitch for 2 studies. Because natural history studies, observational studies where there's no drug, but you're participating, answering questions, wearing devices, using devices from home to measure breathing or muscle. These kind of activities are super important for, number one, to understand the differences in everyone's ALS, but number 2, to understand the biology so that when we start testing drugs, we test drugs that are better and may work more efficiently in your disease. So it's important. So there's this ALS Research Collaborative called ARC through ALS TDI. If you scan the QR code, it'll take you to the website, and you can participate in the study basically from home, from anywhere. Additionally, there's a very large federally funded natural history study called All ALS. And it's led primarily by Dr. Bob Bowser at Barrow Neurological Institute in Phoenix, Arizona, and Dr. James Berry at Mass General Hospital in Boston. But all of us at Columbia and many other principal investigators are involved in this very massive effort. It is probably the largest scalable natural history study. And this is where I'll also state that we're very grateful for federal funding to have helped us get to this level, because we have had trouble scaling up because of research funding and dollars. So federal funding has helped us launch into our community and really catalyze a transformative change by doing this natural history study. It is a study that people with ALS can participate with. You can participate remotely. Asymptomatic individuals at risk for ALS. So if you have a first-degree relative with ALS, you can participate in the prevent aspect. So symptomatics is called assess ALS. The asymptomatic individual is called prevent ALS. And if you are someone who just knows someone with ALS but have no health link to ALS, you can participate as a control. So anyone listening to this webinar right now can participate. Essentially, this is a huge consortium with 35 sites, but the federally funded expanded access programs and All ALS are all synergized so that when we collect samples, the information are coded under a unique identifier. It is all stored under NIH under a BioSend repository. They're all synergized, and it's also intended to synergize with existing repositories, and it's open science and available for clinicians to any clinician, whether they're in ALS or outside of ALS, to obtain those samples and obtain the information to do further research in the future. So this is a snapshot. We're targeting 2,000 participants by 2026. So it's an aggressive, ambitious goal. But the website is all-als.org. And I'll show you a QR code, but there's a lot of innovations being built in. One of the things I think people don't realize is that this study has the ability to incorporate new studies embedded in it. So if I want to look at an exploratory outcome, if I want to add digital technology, which there is already some in there, we will try to do that. So we have been trying to be innovative and try to make sure it's not burdensome for participation. So if you scan the QR code, you'll get to this website. You'll click on whichever one you want to participate in, and you are able to participate. And this is anyone in the 50 states in the United States can participate. So with that, I will say that all ALS and several of the expanded access programs that are federally funded are partnered with what we call the Northeast ALS Consortium. I'm the current co-chair. James Berry is the other co-chair. And the reason why I'm bringing this into the discussion is that this is the clinical trial consortium that tries to enable and make sure sites are clinical trial ready for all of our clinical trials that are coming down the pipeline. Carly Allen and Beverly Smits really lead the team there, and this is our executive operations team. And it is a collaborative effort with many institutions across on the leadership. So these are just a few of the doctors that have really been elected by their peers to be on the executive committee to ensure that we are led in a way. And we have added actually an advisor, a person living with ALS or a caregiver. Lane Oliff was the advisor for the last 2 years. And we really hear from a lot of people in the community, and they guide us in how we take our initiatives. We have an election for a new advisors coming up, so hopefully we'll announce that soon. We have a very broad and diverse scientific advisory board that helps work with investigators or in pharma companies in developing it so that they make sure they include the voice of people living with ALS, people who are at risk of ALS, and trying to make sure we address the needs of the community and bring better drugs forward. So it is a large network. We've kind of branched into international territories, but in doing so, we've also worked very hard in bridging and collaborating with consortiums in Europe, consortiums in Asia, and now bridging to Latin America and Africa. So I'm very proud of the work at NEALS and becoming more of a global force and trying to make sure that everybody in the world gets the care that's standard and get the genetic testing that's standard and shared rights for access to care. NEALS is not just about clinical trials. It's not just about training. One of the beauties is that there are subcommittees focused on different aspects of ALS, but we also have a clinical research learning institute. And I bring it up because we're in a window of opportunity where people can be nominated by their physician to participate in the Clinical Research Institute. We have an annual meeting in October every year. And this is to get trained as what we call a research ambassador. And research ambassadors are people who go out there and try to get the cutting edge information, try to educate their peers, try to do mentorship, try to tell researchers how to do things better. And so there are opportunities to participate in what's called a peace committee, where they can interact with multiple stakeholders. They can develop special projects. We have breakout groups, educational webinars. Sometimes research ambassadors will help us develop the materials and the agenda for our annual meeting. So if you are interested, you can reach out to me or you can reach out to NEALS, and we can tell you the process in trying to do it. We have a limited number of spaces for in-person at the annual meeting, but we do conduct these virtual, and there's multiple places and multiple hospitals and ALS centers that do their own clinical research learning institutes in person. So Pennsylvania, Dr. Terry Hyman Patterson has done one in Philadelphia. We have many of these virtual ones. So there's a lot of opportunities to kind of help out and be a team. Navigating the clinical trial landscape, I just talked for like 30 minutes and probably it was encyclopedic, but I think one of the best tools out there, although NEALS has a website for clinical trials and search, we are partnering and working with ALS TDI on their Trial Navigator tool. And this is actually the team that's there that's working on this is really trying to make sure that it's accurate, it's updated. Clinicaltrials.gov is hard to navigate. And I have to mention also IMALS also has a dashboard as well. They all have their strengths and weaknesses, but it's really important to make sure you know where to look. And hopefully you have an ability to talk to your physician and your healthcare team about things that you're interested in or questions. So I talked about clinical trials, existing therapies, clinical trials, what our pipeline looks like. How can we make drugs better? Well, if we understand the disease better, we could make drugs better. So the Natural History Consortium is really important because we need the samples in large scale to really do the evaluation, to understand targets and understand genetics, genomics, metabolomics, proteomics. All of these information is very important for us. One of the newer kind of focus on trying to understand ALS is understanding the environmental and the gene interactions. So there's 10% or so of individuals that have a gene mutation that explains their ALS, but 90% we don't really have a good understanding of why their disease occurs. And so exploring what we call the exposome, this kind of genetic code that might have some interplay with environmental exposures, is very important. And this has piqued my interest over the last year. This was a publication from Michigan, from Steve Goutman and Ava Feldman. And they've done a lot. And actually, there is a webinar done by Steve Goutman for NEALS. If you go to the NEALS educational webinars, he gave a great talk a few months ago on environmental risks and exposures, what we know, what we need to learn. But recently, I think it was March, there was this kind of cluster in the Alps of ALS. And there were a lot of people investigating why many people over there in a small town developed ALS. And so there is some thought that it could be something related to the mushrooms, but this kind of harkens back to a historical event that occurred in Guam, where the Chamorro people of Guam developed a cluster of ALS and Parkinson's disease, and it was linked to these kind of cycad seeds. And so clearly there's naturally occurring environmental things that may be related to developing disease later. So our understanding of environment and environmental toxins is really important, and boosting that research at this point in time, and also understanding the genetic and environment interactions is going to be very important. Other things, TDP43 is a common protein that's expressed in frontotemporal dementia and ALS. And so the development of a biomarker and target ALS has been very critical in developing one of the new cryptic exons they just announced. But cryptic exons is a biomarker. You might hear it. It might play a very essential role in understanding how an investigational drug is working or not working, but it also may be a biomarker for disease and risk genes. So cryptic exons is kind of like the new exciting topic for biomarkers beyond neurofilament. And then for those interested in the genetics, I talked to you about antisense oligonucleotides, viral vectors, and often you hear in the news about CRISPR gene editing strategies or gene editing strategies. There was just an article on an unrelated disease in an infant that was corrected by gene editing. But CRISPR is being utilized in the lab setting, and it's been applied to what we call human cultured neurons. And it showed that this technology can be applied in that petri dish setting, but really there's going to be a lot of steps. I think a lot of people were excited, especially because it's for C9 or F72, which is the most common gene. But we have a few steps and several years to get through some toxicology to make sure this kind of technology doesn't edit genes that are not supposed to be edited or cause toxic issues. So the folks at UCSF and Professor Claire Cleland is working on these technologies and trying to kind of expedite, thinking of ways to expedite the toxicology so that they can move to clinical trials faster. So that's coming. And then last, I wanted to talk a little bit about technology. I think while we're trying to figure out better drugs, better targets, and try to understand the biology of the disease, I really think that technology in so many ways, so whether that's technology to help with function or whether it's using AI to kind of scrape the internet and get all the up-to-date information on programs, clinical development programs, try to match you to a program. I mean, that's my vision, hopefully. But there's a lot of ways that we can utilize technology to help us. And so bringing these researchers into ALS is really important. Last year, I got a lot of calls during the Olympics. We had a para-athlete carrying a torch with using an exoskeleton. And there are some commercially developed exoskeletons for spinal cord injury. The folks at Stanford and actually at Harvard and also the VA have worked heavily on brain-computer interfaces, or BCIs. And so it's been very exciting technology, especially for those that have difficulty speaking and communicating. And then, of course, the bottom picture is a picture of what we called an iBot back in the day. I've actually seen one of these in real life in 2011 or 2012. And the company went out of business. And I think there are other tech companies that are trying to bring back this kind of iBot wheelchair. So the balancing act it did. So the person that had it really told me that he felt he loved the chair because he liked being up at eye level when he talked to me and the other healthcare providers. He loved the ability of the chair to go upstairs and downstairs, but there was a lot of training that had to be done. But it's based on this gyroscope that keeps the wheels balanced when it climbs stairs or when the wheels stack on top of each other. It's pretty cool. So technology has a lot of potential. One of the potentials is here. I'm going to show you a video. It's from Synchron, which is a company that's based out of New York City. And they're working on ways to create better methods of brain-computer interface. So hopefully this will work. And some of you may have seen it.
Meet Mark. Mark has ALS and can't use his hands. This means he can't use his hands to control a computer or any of his devices. Mark has Synchron's brain-computer interface, also known as a BCI. It sits in a blood vessel on his brain and senses his brain activity. This information is translated into commands and seamlessly sent from the BCI to the digital device he'd like to use. No open brain surgery was required to implant this device. Now, let's watch Mark use his BCI to help control his Apple Vision Pro. Navigating within a Vision Pro screen requires the use of your hands, eyes, and voice. Much like other VR headsets, the user makes selections by looking at what they'd like to engage with and pinching their fingers together to make a selection. The Synchron BCI makes the Vision Pro accessible by bypassing the need for hand gestures and allowing the user to interact with the Vision Pro through a combination of eye tracking and thought-controlled selection. First, Mark's going to send a text. Even though he cannot move his fingers to type on the keyboard, he can use his Synchron BCI to select words or letters by moving the cursor and thinking about moving his hand. Each time a new word populates in the text field, that means Mark chose that word just by thinking about moving his hand. Now let's watch Mark play a game. When wearing the Vision Pro, he can play an immersive game of hands-free solitaire by looking at his card of choice and selecting it by thinking about moving his hand. Finally, let's watch Mark use the Vision Pro for an immersive entertainment experience on Apple TV. It's as simple as navigating with his eyes and selecting the movie he'd like to watch. Hands and voice free. By integrating the Vision Pro directly with his Synchron BCI, Mark can enjoy immersive experiences, accomplish tasks, and connect with his digital environment in new ways.
So I think that's just one example of how technology can really change someone's quality of life. So in Mark's case, he has trouble speaking and he has no function in his hand, and the implanted transmitter acts almost like a Bluetooth and can connect to the device and help. So traditional brain-computer interfaces used electrodes placed on the brain or sometimes implanted and required surgical implant to do it so that it could pick up the electrical signals from the brain when you think about something. But this is a very novel technology that doesn't require open brain surgery. Now, this slide that you're looking at is a project from the engineering lab here at Columbia University. They've collaborated with all of us in the ALS Center to kind of think about ways that we can improve supporting neck weakness. Now, there's exoskeletons I showed you of the legs. There are some beta exoskeletons of the arms. And of course, many of you might be familiar with the OB device, which is not an exoskeleton. It's a robotic arm that helps with feeding. But there's been an unmet need for support of the neck. A lot of what we use is traditional like cervical cord, spinal injury collars. So they're very rigid, they're very stiff. They sometimes are constricting. Soft collars are not supportive enough. And so this is actually a 3D printed brace that is developed, and all the joint and support is really on the shoulder and head rather than the neck. And the student that's actually demonstrating it actually worked with the engineering mentor to develop these spherical and rotational joints so that if you use these joints, you can get full rotation of the head in every direction. The only restriction is really coming all the way back beyond neutral position for the head. And so the first one, the orange one on the left, is the passive brace, which we used for measurement. But this middle video is someone using a joystick to help move if you have no ability to move the neck. And then there's adjustable passive support where if you have some strength in the neck, but not full, just a little motion can get it engaged. And we're doing a second version of this by using eye trackers. So there's eye tracker glasses that someone can wear, and based on movement of the eyes, they can move their neck up and down. And mainly what we want to do is make sure it doesn't cause pain, discomfort, and make sure we're not activating muscles that we don't intend to and cause neck fatigue. So that's ongoing and still being developed over at the School of Engineering at Columbia. And last, I think, AI and technology is really kind of jumped far levels. So there's a lot of people I noticed in clinic have already discovered 11 labs for voice generation. Team Gleason has been very helpful in helping with speech technologies. There's acapella that can recreate your own voice. So there's a lot of technology that has been invested into generating your own authentic voice. There's a new foundation, the Scott Morgan Foundation, that's trying to develop visual avatars so that people can use 11 labs or other types of voice generating technology along with eye gaze to create a realistic avatar to communicate and interact in real time. And so this is Eimer Barrett. He's actually part of the development here at Scott Morgan, and I'll show you. He explains this technology.
Hello, I'm Eimer Barrett, living with ALS, a progressive neurological disease gradually robbing me of my ability to move and speak. Yet, as you can see, ALS hasn't silenced my mind or determination to communicate. The voice you're hearing and the expressions you're seeing, that's all me, preserved and animated by AI. This isn't just mimicry. My hyper-realistic avatar by DID is an extension of me, maintaining my personality and mannerisms. My 11 Labs voice preserves my natural speech patterns, while an iRespond eye tracker lets me control a custom-designed keyboard. That.
I think this is where, the technology is there. It's being refined both for speech and because sometimes when you do the first run, even with 11 Labs, sometimes you don't get quite your accent or quite your voice, but there's ways to refine it over time. And so this is the Scott Morgan Foundation. This is actually from their website, and this is the front homepage. So what I think is what was a tough year, 2024 was a tough year for us in ALS, and we still have a tough road ahead, but I think the future of ALS is getting brighter. We have been fortunate to receive the federal fundings through NIH and through DOD that has really catalyzed the field since 2022. And we really need to make sure everyone in government knows that we really want that continued funding to support the research, because that will help us get us there faster. Foundations and industry have done their share and also continue to support the pipeline despite all of the challenges we had last year with therapies. And so one of the things that we've learned is that we really do need to diagnose ALS faster and earlier and make sure we have access. So that includes developing our workforce. We really want to make sure that genetic testing is available to everybody if they choose to do so. And so it's really important to be screened for SOD1 since we have a therapy that's approved. As you noticed in the clinical trial pipeline when I presented, FUS, C9orf72, TAR-DBP, and some even ultra-rare mutations may have experimental therapies, so it's important to know that. But it also has impact to family members that are not affected, and it provides information for them and their risks. And it also provides tools for people who might be planning families. I talk to a lot of young women who are planning to have children and they don't have ALS, and they do pre-implantation genetic screening. and do in vitro fertilization to have a healthy baby that they know and reassured that does not carry the mutation. So there's a lot of things that people can do proactively if they have the knowledge. We are learning more about biomarkers. Neurofilament, everybody asks, and now we can clinically test, but it is difficult to interpret one measure at one point in time. We do have to understand how it changes over time, and different assays and different labs have different cutoffs, and so sometimes that can be confusing. So we are learning, and the more we test and the more we get more information, like in studies like ARC from ALS TDI and also from all ALS, we will have a better way to predict what to expect for you in the future. Studying environmental exposures, I think, has raised on my priority list and also on the priority list of our research community. So understanding and doing epidemiological studies. So ALS United is very much involved with New York State to try to see if we can get a state registry. There are several states in the United States that have a registry for ALS. And understanding lifestyle, diet, environment risk factors, occupational risk factors that can contribute to developing ALS. We want to make sure that we're including people living with ALS and people at risk when we're thinking about clinical trials or developing potential therapies. And we have to make sure to evaluate people who are at risk as well, and who would volunteer and want to get tested and provide their samples. This is a valuable cohort that can really help us understand how to prevent disease in the future. And that's the example I gave with the SOD1 study called ATLAS, where people are willing, after they know that their loved one has been diagnosed with ALS, they get tested, and if they carry the SOD1 but have not yet developed ALS, for SOD1 individuals, carriers, they can participate and use the drug that's available to prevent ALS. We continue to innovate in clinical trials. I took a lot of the slides out, but Silence ALS is an example of these kind of personalized kind of clinical trials, but we're trying to be more precise in how we target therapy. So understanding the biomarker and trying to target therapies according to what might be more prevalent in your disease. So not all ALS is the same. Somebody might have a lot of oxidative stress. Somebody else might have a lot of neuroinflammation. So getting those biomarkers, understanding them are really important. And even looking at neurofilament and seeing if whatever you're doing is lowering neurofilament could be helpful over time. We want to make sure that if there is a big pipeline that I showed you from phase 1, phase 2, that we test them rapidly. And so the platform trial does allow us to kind of test things in a more efficient manner with less number of people and less time. And most importantly, I think with all ALS and natural history studies, we really want to make sure that even the clinical trials that are completed, they all go into a repository so that we can promote open science, collaboration, and sharing. I really am privileged to work in this community, and in 20 years, I've seen a dramatic change in how everyone collaborates, shares, and tries to really work together and use our strength and numbers to really do great advocacy, improve clinical care, and improve the ways we conduct research. So I am very hopeful, and I do believe the future is bright and that we will get there. And hopefully with all of the work that is being done, it's not just about the researchers and the scientists and the funders, it's also about people living with ALS and their families and friends and the community all coming together. So with that, I'll conclude for now and see if we have questions, which I'm sure there are.
Thank you, Dr. Andrews. You have a very special way of giving clarity to a very complex topic. So thank you so much. There are some questions. First one is, what are your thoughts about the University of Michigan statement that the rate of ALS is expected to rise by 10 to 20% in the next decade?
Ooh, that's a hot topic. You know, it is true, it's going to happen. And so here's the situation. It's not just that. So as time goes on, just standard of healthcare in the United States and globally will improve and people will live longer. If you live longer, as you age, your chance of developing ALS increases. So if the population ages and we have more people living longer, we will have increased incidence. However, you know, after this little story about the Alps and this little cluster of people with the mushrooms, I went back and looked at Guam, and for me, I wasn't there, so I had to reread the history. And, I think that we have to really understand the environmental exposures. So, there is a publication, besides University of Michigan, there is a publication that globally will have a 70% increase globally for ALS by 2040, for a variety of reasons, but a lot of the times the aging population is cited. But it's not just for ALS. There's going to be an increased incidence of Parkinson's disease. There's going to be an increased incidence of Alzheimer's disease. And if you look at all these diseases, like about 10 to 15%, 15 to 20% are genetic, and a majority of these people don't carry a mutation for these neurodegenerative disorders. So I had the privilege of being on a panel with a Parkinson's disease specialist who specializes on environmental toxins. And he was saying how the numbers are increasing. We can't attribute it completely to the aging population. We really have to understand environmental exposures. And we do know of exposures besides the whole cycad seed BMA toxicity. There are, military service in the United States is a risk factor, right? So there are things about lifestyle, occupation, and environment that do contribute. And there is a change, when our society becomes industrialized, there are things that occur that could possibly be contributing. That's why the research about environmental toxins, environmental and gene interaction is going to be very critical, not just for ALS, but for all the neurodegenerative disorders.
So interesting.
Yeah.
Another question. How long are the B12 injections recommended for?
Yeah, I get that question a lot. The B12 injections, just so everybody knows, the injections that you get from like your primary care doctor, if you're B12 deficient, is an injection you get monthly, and it's cyanocobalamin. It's different. And the oral formulation could be also different. Methylcobalamin is a formulation of B12, but it's just methylated. And so like it's very red. So if you get an injection, it's going to turn your urine red. So don't be alarmed. It's an injection that's done twice a week. And the study was 4 months. But the thing is, it's meant for neuroprotection. So there is no clear end date. When I work with people living with ALS, we'll talk about the pros and cons, talk about whether they are needle scared, needle phobic, and how we would manage it, and talk about the cost, because that's also a risk that you have to talk about, this financial and health related risks. But we also talk about when we would consider stopping it. And there's no clear, I'm going to tell you right now, the short answer is there's no clear answer as to how long you're supposed to do it. But when I talk to somebody, I will talk about, what threshold, if you feel like you're not, if it's not doing anything, or you feel like you hit a point where it doesn't seem like it's worth continuing injections and paying money, when you've lost a function, because B12 is not going to help you regain that function. It's supposed to help stop progression or mitigate, slow down progression, and preserve function for as long as possible. So I think it's a continuing conversation, and you kind of have to look at the side effects, and actually it's tolerated very well. I think most of the time people have a hard time with just injecting at home.
Thank you.
Yeah.
There's also a question about getting one's genes identified. This person is saying she can't come to Columbia, so how could she get her genes identified?
That is a good question. If this individual is asymptomatic, I would strongly recommend, and if that individual, whoever you are, have someone, if you're asymptomatic and you have someone related to you that has ALS, then I would strongly suggest linking to the All ALS program, where you can be screened under a research protocol. And that way you can be evaluated, and it'll be all kind of separate from a clinical protocol, and you can do it remotely. You don't have to come to a center. The other thing is that WashU, Columbia, I guess it does require some genetic counts. You do need, if you're symptomatic, you should be seen somewhere, and they should be able to get genetic screening for you. But the most important part, whether you want to get tested or not, in whichever way you want, is that you have to, I think it's important to have access to a genetic counselor. I think that's the hardest part for people to get screened.
Another question, how do I choose the right clinical trial for me?
Oh, that's the hardest question ever, because it used to be, there's one trial, like 20 years ago, I just had one trial that would start, that would end, and there would just be another trial in sequence. And I think one of the benefits now is that there's multiple trials, there's clinical trials and there's expanded access. And so sometimes I have a person come in, like I had clinic yesterday, and I had to talk about 2, 3 things, and I was excited to present multiple things, but I confused the person. So I had a woman come in who helped me think things through from their perspective. So a couple of things you want to think about when you think about the clinical trial. Number one, you have to ask, what phase of development is this product in? So is it phase 1, where there's high risk and not a lot of knowledge about safety? Are you willing to take that risk? And some people are. And some people say, you know what, I want to wait for something that has some safety data in humans. So that's usually phase 2 or phase 3. Some people want a lot of assurance. And so that means that a product would be in phase 3 because there's some preliminary signal that it helps. The second question is there a placebo? And what's my chances of placebo? And typically in the informed consent that you get, the investigator should tell you if there's a placebo arm and what your chances are in very plain English, not like some statistical odds. Whether it's 50% like a flip of a coin, or is it like 1 out of 4 chance, 25%. So they should be able to explain. And some people are okay with that. Some people will say, okay, yeah, I'm willing to go on take that chance on placebo, because after the commitment, I do get people who participated in the clinical trial get first access to the drug in the open label extension. So sometimes people are willing to participate in a placebo control trial. Some people will say, no, I don't want to do that. And so sometimes an open label clinical trial, open label means there's no control arm, or an expanded access, if you qualify, would be the choice. The third is the safety profile of the compound. And so it's not just the safety, like what are the bad things that happened with this? And that's usually in the informed consent, but it's also, what's your medical history? If this is a drug that raises blood sugar, but is supposed to help your motor neuron, but you have diabetes, that's not the drug for you. Or if that drug is supposed to help slow down disease progression and help the survival of the motor neurons, but it causes cardiac arrhythmia, and you have a history of cardiac arrhythmia, that's not the drug for you. So your medical history plus the side effects of the drugs should be, so the adverse event risk profile. And the last 2 things I would think about is, how often do I have to go to the center? How far is the center? Like, how many trips do I have to make? How long do I have to participate? And what's the route of administration? Is it a pill I can take at home? Is it something I have to inject myself? Do I do it at home, or do I have to be at the center? Is it IV? Is it home infusion or at the center? Is it intraspinal? And there are going to be some, I told you about a stem cell study where they're injecting, it's a surgical procedure, they inject into your brain. So the route of administration usually dictates how people want to participate.
So much to think about.
Yeah.
There is a follow-up question regarding B12. To clarify, is it okay for someone to exceed 1 year on B12? Let's say 1.5 to 2 years, maybe.
Yeah, I mean, I've had patients on it. Usually we monitor the B12 and folate levels. You want to make sure the other measures that are surround B12 are okay. If they're off, sometimes we do take people off if their B12 is way above 1,000. But normally the B12 that you don't use the excess, you usually urinate out. It doesn't accumulate.
Thank
That's a
you.
good question. It's a tougher question than it seems, because I don't have answers. I wish I had more answers.
We'll do. And then what do you think about the National Registry and promoting research?
Yeah, the National Registry is the CDC registry, and it's a little bit different than some of these state registries. But one of the benefits of the National Registry is that it's been around for a while. And it is where we're getting our incidence and prevalence numbers, although it's not perfect per se. And they do have a biorepository where people are giving samples. And so right now, because there's 20,000 or so individuals in there, and there's historical data, it is kind of a valuable asset at this point. I mean, one of the criticisms it's received is that, it's not mandatory for everyone to be registered in the CDC. So we're missing individuals, and it doesn't require the physician to enter data. So you have to be motivated, and everyone who gets diagnosed with ALS and their families can tell you 100%. that it's like overwhelming. The whole process is overwhelming. The last thing they want me to do is give them something else to do, you know? And so they're tasked with, going on the website, registering and putting their information in and giving the samples. So I think that's one of the downsides or criticisms. Ideally, if there was a mandatory registry where either physicians have to report it and as long as we protect individuals who are diagnosed, because we've seen this in the HIV era, right? Where people were discriminated against for whatever reasons with these mandatory reporting of cases. But it's important to understand, I wish every state had a mandatory registry. We did it the same. It's very important to make sure we're doing it in synchrony. And we can see clusters in certain areas and we could see exposures. And if we're collecting similar lifestyle and diet and occupational hazards, we could see links across these. So I think the National Registry brings value in that way of bringing some of the information. But I think if we make it completely reportable, and if it has to be state by state rather than a federal function, I think that would help us in understanding more about epidemiology and environmental factors that cause ALS.
Let's see, I think there's one more. Let's see, I believe one of the advocacy items is to make ALS a reportable disease. I guess that's a comment.
Yeah, and I know that New York State and advocacy have worked together to try to see. New Jersey has had this historically. And this is unfounded what I'm about to say and controversial, but might as well make it a little spicy right at the end. For us New Yorkers, I'm born and raised in New York. I was born in the Bronx. I live here, and I'm a New Yorker when I don't have my white coat on. So I think it's important. There's a lot of things that's happened in New York that could play a role in ALS, and one of those things that I always wonder about is is the whole 9/11 event and its relationship. Just yesterday, I had another person tell me that they were working in the 9/11 cleanup site. And they have a history of cancer, and now they have ALS. And I have a series of individuals that were NYPD, FDNY, or people who worked in that area who volunteered. And there probably is something to that. And I kind of wish we had a New York State registry for this to kind of look specifically. And it's hard for nationally for people to take interest because it's a New York State issue. But I do think there's something there that needs to be investigated. So that's just an example. Not going to touch COVID. So.
That pretty much concludes the questions that we have for today. So again, thank you so much for this clear and comprehensive summary, Dr. Andrews, and thank you for being here and giving us this time.
Yeah, I appreciate the opportunity. And, of course, I always take feedback. So anyone out there, if you want to give me feedback, feel free. I love it. So, and you help in the direction of scientific research. So thank you. Thank you again to ALS United for having me.
Thank you. So for those in the audience, the next presentation's at 1:30. So please consider joining us. It's about perspectives of living with ALS. So thank you.